Good afternoon, guys. Good afternoon, Humera. Good afternoon, Rafi. Okay. Okay. So we will start, and the others, if they want to join later, they can join. Okay. So today we will continue the biology. We will speak about the epithelial tissues and their kinds. So we have the simple squamous, the simple cuboidal, the columnar, the stratified, the stratified cuboidal, and the pseudostratified, and at the end we have the transition. These are the different shapes and different kinds of the epithelial tissues that we have in our body. The simple squamous tissue is a kind of tissue which is one of the simplest kinds of the epithelial tissues, which protects and helps in protection, helps in secretion, helps in absorption, and helps in sensation. So it has so much work, functions like secretion, okay, secretion, absorption, protection, etc. One of the simplest squamous examples is the alveoli, which contains epithelial simple squamous epithelial tissue, which is at the end of the lung. The simple cuboidal, it looks like this. Looks like a cube. This is simple cuboidal, and this one helps in absorbing fluid. If you see, it looks somehow thick since it's a cube, so it absorbs easier and more fluid than the simple squamous. The columnar will be looking like this. This helps in absorbing also like the simple cuboidal, so absorbing fluid and in secreting HCl or enzymes. So also in absorbing and secretion. Okay. We have the stratified, which is one of the biggest epithelial tissues that looks much bigger than the others. Looks like this. Okay. And which has a bigger role than the columnar and the simple cuboidal since it have bigger structure, so the absorbing material is much higher. The stratified cuboidal is more like stratified one, so it will be looking like this, like a cube since it's a cuboidal, and it looks like this. also helps in absorption. And then we have the pseudostratified, which is like this. Of course, more than one. And at the end, we have the transitional, which is one of the most complex epithelial tissues, which looks like this. Okay. So what you should know about them is the alveoli is simple cutaneous, squamous. You should know that we have the different kinds, and each one of them has a kind of role, which all of them have is the absorption, secretion, protection, and sensation. These are the epithelial tissues. Now we're going to speak about the connective tissues that we have in our body. So, the connective tissues are the cartilage, the bone, blood, chyme. These are the connective tissues that we have in our body. They help, these connective tissues has a function in support, in storage, in binding, and different functions. Okay, so we spoke about the epithelial tissues, we spoke about the connective tissues. Now, before we start with the cell sizes, I want to demonstrate something. Okay, yeah. So I want to speak sometime about the cartilage before we continue. So the cartilage. The cartilage is that piece which is on the bone. Say this the bone. It has a piece here. This piece is called the cartilage, the protection around this bone head. We have different types of cartilage. We have the hyaline cartilage. We have the elastic cartilage. And we have the fibrocartilage. The hyaline cartilage, as an example, it's a kind of cartilage. The normal cartilage is the hyaline cartilage. This is the hyaline cartilage, which surrounds the head of the bone. And as an example, it exists in the epiphyses. So, okay, and nasal. Trachea and the ribs, from the ribs to sternum. However, the elastic cartilage have a very high amount of elastin. As an example, in the ear epiglottis. Okay. And the fibrocartilage is in discs and meninges. Let's try to draw the anatomy. Second, yeah. The anatomy of this cartilage. So, let's say this is the bone, okay? We have here This is the cartilage. Here we have the epiphyseal line. This is the hyaline cartilage. Okay, here we're gonna have the red bone marrow. And here we're gonna have the yellow bone marrow. Here we will have the nutrient foramen. And here is the periosteum. And here we have the contact bond. The contact bond. Here blood vessels. Okay, so this is like a kind of way to see how the bone looks like with the cartilage. What happens in the bone marrow? The bone marrow is the place of maturation and the growth and the secretion of B cells and their maturation, and for the secretion of T cells also, the T lymphocytes and the B lymphocytes, which helps in the immune system. Now let's speak about the things which are the fibroblast and the fibroblasts, they lay down bone. Fibroblasts, they break down bone. This also for memorizing this definition. Okay, now we were speaking about the connective tissue. We spoke about the cartilage. Let us draw how is the shape of this connective tissue? How does it look like? So here we have, let's say, the epidermis. Then so here we're gonna have the melanocytes. What does these melanocytes do? They secrete the melanin to give the color of the skin. Here we're gonna have the Langerhans cell. And here we will have the basal layer. And here the small unit at the end is called the Merkel cell. Okay, so the connective tissue proper, okay, is we have the melanocytes, and of course on the epidermis, what do we have on the epidermis? Let's see if one of you will know. What do we have on the epidermis? What do we have on the epidermis?
Your voice is low.
Hmm? Let's see if someone responded. Okay, I'm asking. Oh, you cannot hear me, guys?
Yes, we can hear you now.
Okay, perfect. So what do we have on the epidermis? Here, let's say we said that this is the epidermis. We have dead keratinocytes. Okay. So, dead keratinocytes. The Langerhans cells, what are they? Langerhans cells, or other name is Langerhans cells, are a kind or a specialized type of skin immune cells that play an important role in the skin's protective immune system by helping for activation of the TK cells or cytotoxic cells or T cell mediated responses. So the Langerhans cells, they help in the immune response. Okay, so we spoke about the fibroblasts and the fibroclasts. We said that the fibroblasts, they lay down bone. However, the fibroclasts, they break down bone. How does the fibroblasts look like inside this connective tissue? Okay, so let's say here we have, let's draw. Okay, so here we're gonna have the lymphocyte in this tissue. Here we will have free macrophage inside. And we will also have collagen here, blood vessels here. And the fibroblasts will be sitting here in this way. They look like this. These are the fibroblasts. And here we will have a plasma cell. So inside this connective tissue, we're gonna have different types and different kinds of immune cell types. Here we have a ground substance. Okay. Before we continue, I want to give kind of small note. So, what is cystic fibrosis? One of the most diseases which are asked in the IMAT. Cystic fibrosis is a disease or is a mutation to a gene that codes to a protein exactly, bravo, to a protein in the plasma membrane, which is responsible to pump Cl minus ions. An effect of this cystic fibrosis, we have accumulation of mucus, or an effect of this mutation, we have an accumulation of mucus, and when this accumulation of mucus happens, cystic fibrosis will happen. Okay, so it's a kind of mutation to a gene that codes to a protein in the plasma membrane in which it's responsible to pump Cl- ions. When this mutation happens, so much mucus will be accumulating. This accumulation of mucus will lead to cystic fibrosis. Okay. Now, we're going to speak about the cell theory. So before we continue, we have to speak about Robert Hooke. Robert Hooke had 3 laws. He used to be an engineer and a scientist who invented the microscope. This Robert Hooke, he created or he made the 3 laws in order for us to know the cell theory. So number 1 law of Robert Hooke, it's all organisms are made of one or more cells. Number 2, he said the cell is the basic unit. The cell is the basic structure and functional unit of all organisms. And 3, he said cells arise only from division of 3 existing cells. What does this mean? He's speaking here about the sexual and the asexual reproduction. Of course, the sexual reproduction needs a male and a female to be able to fertilize this egg and fertilize that for the zygote to be created. However, the asexual reproduction only needs one female or one male to replicate or to for mitosis to happen in this cell. And of course, all of these cells are identical to the dad cell or to the mom cell. However, we have a higher genetic diversity with meiosis. Now let's speak about the cell size. So we have 1. O2 molecule is 0.07 nanometer. DNA helix is 2 nanometers. Cell membrane, which is normally the lipidic layer, is 9 nanometers. HIV virus plus the influenza is 100 nanometers. E. coli bacteria is like the mitochondria from 1 until 5 micrometers. Human egg is 100 micrometers. 7. We have plant cell is 10 to 100 micrometers. Vacuoles is 1 micrometer. What else? Animal cell is 5 to 30 micrometer. Micro is times 10 power minus 6. Nano is times 10 power minus 9. Mitochondrial cell is 1 to 5 micrometer. It's the same as E. coli. Okay, another one. Where should I write it? Staphylococcus bacteria, 30 millimeter. Also, red blood cell is 6 to 8 micrometer. Okay. White blood cell 12 to 17 micrometer. Onion cell, you should just know that it is much bigger than red blood cell and white blood cell. Onion cell is measured in millimeter. So let's try to first take screenshot of this before we continue. Okay, now let's try to do some comparisons. E. coli bacteria. Okay, as I said, it's 1 to 5 micrometer. So we have E. coli, and then we have red blood cell, and then we have onion. Okay. Plant cell is bigger than animal cell. It has a vacuole. It has so much bigger organelles. Both of them are measured in micrometer. Virus are slightly smaller than bacteria. Bacteria smaller than animal or plant cell. Okay. Red blood cells can live about 120 days. White blood cells can live from 12 to 20 days. Women blood in her body 4 to 5 milliliter. However, the men, no, liter, I guess. Liter. Men from 5 to 6 liter. A human bladder can hold 600. Let me make sure about this. Just a second. Yeah, from 400 to 600 milliliter. Here, liter, nothing. Okay. What else? Let me see if there's anything additional. Okay. I guess that's all. Yeah, before we continue, there's also something. So, contents of saliva. Saliva contains 99% water, contains electrolytes like mucus, like IgA, immunoglobulin, enzymes, lysozymes, the ones that we spoke about them in the slides. Okay. The pH where pepsin can work. Two. Cytosolic enzymes pH is 7.2, almost neutral. Okay, perfect. Now, since we already entered digestion, let's speak about the digestive system. Yeah, antibody, exactly. So let's speak some time about the digestive system. So, of course, the digestive system has the two properties, which are the mechanical and the chemical digestion. The mechanical means movement, means pushing, means contraction, means relaxation. However, the chemical means the enzymes, means the juices that are secreted to facilitate this digestion. So, let's say from the mechanical part of the digestion, you should memorize two definitions. So we have the regurgitation, and we have the peristalsis. The regurgitation, as the definition, is the forceful contractions of abdominal smooth muscle backward flow. However, the peristalsis is the series of muscle contraction propagating the digestive tract. Usually, who has this regurgitation and this peristalsis are the stomach, since the stomach is the most organ in the body, in the digestive system, which helps in breaking down these molecules into very small molecules, which are called the nutrients. It makes a very high mechanical movement by churning, and this churning will lead to a high regurgitation and a high peristalsis. Who makes this peristalsis? Which kind of muscle? The smooth muscle. Okay, okay, so another notes you should take in the digestive system are, so the liver, it produces bile, produces bile salts. The gallbladder, it stores bile and it releases bile. The pancreas, it secretes pancreatic juice. Okay, and secretes from from the endocrine. Not just here is the exocrine function of the pancreas. However, the endocrine one, it secretes the hormones, which are the glucagon and insulin. Okay. However, the pancreas, it secretes the glucagon and the insulin, and everything starts from the pancreas and stays in the pancreas. The glucagon and insulin starts in the pancreas and stays in the pancreas. We're gonna speak about this. Just see when. Yeah, soon. Soon. Perfect. Okay, let's continue. So, what happens in the digestive system? First, we start with the mouth. The food enters the mouth. Salivary amylase, the saliva is there to break down the starch, and then it's broken down and swallowed, and then it goes to the esophagus, and then from the esophagus it will reach the stomach. From the stomach it will go to the duodenum. Duodenum is the number one station to reach the small intestine. In the small intestine, the absorption will happen because the only transportation that we have in the small intestine for circulation of these nutrients all around our body organs is the blood. It will absorb these nutrients. However, everything which is waste, it will go. Yeah, I'm not writing, don't worry. So it will go to the large intestine. And then the large intestine will try its best to push for them to go out through the anus. Most of the juices that are produced by the pancreas, that are produced by the liver, the enzymes, the lipases, the gastric juice, all of these juices, most of them work on a very acidic medium. What organ in the body has a very acidic medium is the stomach. And the stomach has a very thick layer to protect it from burning. When the stomach has this highly acidic medium, very low pH, what happens? It needs a thick layer to be able to protect it from burning. Because if we don't have this layer, this organ will burn. However, the pancreas, it still produces a very low pH juice, which is the pancreatic juice. But the pancreas does not have this thick layer like the stomach. So what happens in the pancreas? The pancreas try to produce another kind of enzymes and another kind of secretions which are called the lysogens. These, as an example, the trypsinogens, they try to balance the pH of the pancreas because it's producing this juice which is really acidic. So in order the pancreas not to be burned by this acidic juice, since it does not have a thick layer, it produces this trypsinogen and not lysogens. They are called zymogens. So okay. So as we said that this pancreas does not have a thick layer, it has a thin layer. So it needs the zymogens to be able to function in a balanced medium. What happens in the stomach? In the stomach, we're gonna have so much juices, so much enzymes working all together with the movement, which is the churning movement, and the peristalsis and the regurgitation, and all of these mechanical and the chemical movements to be able to digest in the perfect way for these nutrients to reach our body and for the waste to go out from the large intestine. However, in the large intestine, this water, this nutrients will be already absorbed. However, everything which is waste, it will be going to the large intestine, and then it will leave the body by the movement, by mechanical movement. Okay, perfect. What are you saying? Sayed was saying something. Let me see. Is she right? No, no, I'm not writing. Do you want me to write? Because I guess this is easy, you know. But if you want, I can write. I will write. Don't worry. Perfect. Okay, so we spoke about, so we said we have mechanical and we have chemical. Okay, it will go from the mouth to the esophagus, then stomach, small intestine, large intestine, then here we're gonna have the anus. What happens? The food will go from here, reaching here, here, very huge mechanical and very huge chemical reactions will happen. And then they will go to the small intestine for the absorption of nutrients by the blood will happen. And then they will go to the large intestine. We were saying that in the pancreas, we have the zymogens, which help balance the medium for the pancreas not to burn, because the pancreas is secreting so much juice, and this juice is really acidic. So if it stays in the pancreas and the pancreas does not have a thick layer like the stomach, so then it will burn. Other than this, I want to try to show you the anatomy of the large intestine. So, of course, looks like this. Then here we have the anus. So, of course, we have one ascending, one descending colons. So the ascending colon is this one. This is going up. This is called the transverse colon. This is descending. Okay. Here we have this segment here. It's called the rostrum. Rostrum. Okay. We have here the cecum. Cecum. And we have here the appendix. It's like a small here. What else we have here? Yeah, here the one before the anus is called the sigmoid colon. The rectum. And what you should know also, I think transverse, ascending, descending, cecum. Yeah, there's, let's go inside somehow. This is the cecum. Okay. If you go inside this, like after the cecum, if you go inside to the inside, you will have something called the ileum. So it starts with the appendix. This is the appendix. Then the cecum, this big unit. If you go to the inside, it's the ileum, ascending column, transverse column, descending column, sigmoid column, rectum, and anus. The sigmoid is this one. It's from here, reaching here. This is sigmoid. Okay. And this here called the anal canal. So appendix, cecum, ileum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, anus. Okay. Now, perfect. Now, I want, before we continue, I remember we spoke one time about the lytic and the lysogenic cycle. You remember, guys, what were they? Are disorders? Yeah, I will give you some diseases which are important in some systems. However, the ones which are not important, I won't give it to you. So I will give you them, don't worry. So you remember the lytic and the lysogenic. So how does this virus or this bacteria know if it will undergo the lytic cycle or the lysogenic cycle? How do you think, guys? Do you have any answer, Sayed? For the time. Yeah, how does this lysogenic endolytic cycle, how does the bacteria or the virus know, okay, now, like I should undergo a lysogenic cycle. Now I will go and to the DNA and infect this DNA to kill this infected cell. Or now I will go and replicate myself, me as a virus. I will go sit on the side of the hosted cell and replicate. How does it know? Should I enter the DNA or should I not? Or why should I replicate on the side or go inside the nucleus? How do you think they know? What is the thing that happens to choose between the lytic and the lysogenic cycle for replication of viruses? We spoke about from like 2 days, 2 days or 3 days. I don't remember, to be honest. I Are you watching the sessions or you studying with us? Because if you're watching, I don't think you reached. Okay, so the virus knows that he should undergo the lytic or the lysogenic depending on the nutrients. If there were nutrients, it will undergo the lytic cycle. If no nutrients, it will undergo the lysogenic cycle. Okay, it's also something you should know. Okay, we spoke about cell size. Good. Now, okay. Now before I continue, let me just make sure I have them. Because I want to give you the diseases that might come on that. So let me just see if I have the list. Since he asked about them. I don't have the list. By tomorrow, I will make the list. I will give you the list tomorrow, okay? But if you want, I can speak about other diseases. Let me speak about them. So let's go for diseases that might come on the IMAT. So according, if we go to the heart, you might see the congenital heart disease, myocardial infarction, cardiac arrest, heart failure. This is for the heart. For the blood vessels, you have aneurysm, you have hypertension, strokes, thrombosis. Thrombosis is mainly blood clots, and arterial polymerosis. This for blood vessels. Now, for blood, like blood tissue, the blood as a tissue disease, so blood disease, you have iron deficiency, you have anemia, aplastic anemia. You have so much types of anemia, to be honest. Sickle cell anemia. The sickle cell anemia is the one that has the alpha beta, and then one of this will be mutated, and then it lost its structure. This means it's lost its function, and then this anemia happened, this sickle cell. If you remember, we spoke about it at the beginning of the session. You might have the leukemia, means the very high white blood cells in blood. You might have hemophilia. Okay. These are mainly the diseases. There is another diseases which are respiratory. Let's talk about them. So, respiratory diseases. We have two kinds of diseases. They can be restrictive or obstructive. Usually, the restrictive diseases, they have a low lung volume. However, the obstructive, they have low airflow. Okay. In restrictive, you have a difficulty in inhalation. So difficulty in inhalation. However, the obstructive, you have the shortness of breath. Here you will have stiffness. Here you will have high exhaling, trying to get air from outside. You don't have a good airflow. You're not able to get the oxygen which is outside. So you try more to exhale. Like asthma, you know? Like people who have asthma, they know about this. Or COPD. However, here in the restrictive, like interstitial disease, scoliosis, which is marked obesity. Usually obese people has the restrictive one. Okay, another kind of lung disease. We have the emphysema. Let me write it. Okay, which you have a destruction of the alveoli in the emphysema, and then this will make problems in the lungs. Usually, these are problems in the lungs. Okay. Okay. Other than this, let me see if I have any other diseases. Mm, no, these are all the diseases. I think there are still, but in genetics, I will give them that now. There are still, but the ones that I will give, they are according to genetics, not according to body system. Okay? Or if you want, I can give them now. Let's do them. Since we already speak about diseases, so we don't repeat later. So let's speak about genetics. You should memorize these. So you have the autosomal linked diseases. And you have the autosomal dominant. And you have the autosomal linked recessive. So the autosomal linked dominant are Huntington's chorea, free earlobes, achondroplasia, which is also called dwarfism. You have ear balding, no, early balding, sorry, early balding. This usually happens in male more than female. You have curly hair and progeria, which is premature aging. We have and dactyly, which is digits. The recessive autosomal linked are albinism, attached earlobes. The free is autosomal linked dominant. The attached ones are recessive. Attached earlobes. Cystic fibrosis, which we just spoke about. Sickle cell anemia, which we also spoke about, is recessive, and the Tay-Sachs. Okay. However, you have one. However, there's one which we spoke about in the blood. We said that we have hemophilia, if you remember. The last one we spoke about in the blood diseases, which is hemophilia. Hemophilia, guys, is sex-linked recessive disease. Okay. Okay. Yeah, curly hair is a disease. Don't be shocked. Okay. Now we now I want to go back some time for photosynthesis. So I want to go back. If you remember, we spoke about glycolysis and fermentation yesterday. Now I just want to finish bioenergetics. So by the photosynthesis, and then we continue. So we spoke about photosynthesis and we drew this cycle, which is here. Here we have the light-dependent reactions. And here we have the light-independent reactions. Here it will use CO2, produce glucose, and then use H2O to produce oxygen. This happens in the thyroid membrane, and this happens in the stroma. The light dependent in the thyroid, however, the independent, or the Calvin cycle, let's say, in this stroma. Here, the light dependent uses a photon, so this means the excitation. So the excitation happens in the thyroid, however, the Calvin cycle or the independent reaction happens in the stroma. Let's now speak about this excitation in details and this Calvin cycle in details. Okay? So let's first start with the light-dependent reactions. So let me just draw the photosynthetic apparatus or system. Okay. Okay. So it looks like this. Let's say this is the chloroplast. Okay. Here we said that the chloroplast has outer membrane and inner membrane. So this is the outer membrane. Here we have the inner membrane. Looks like this inside. These, if you zoom in, you'll find here the thylakoids. So, thylakoids. Here in this thylakoids, the light-dependent reactions happen. Okay? Let's say a group of these thylakoids is called the granum. And from one thylakoid to connect to the another, this is called the lumina. Connection between a granum and another. So lumina is this. As a definition, it's the connection between a granum and another. A granum is a group of thylakoids. Here, a granum and another. We have here the stroma. And in this stroma, what happens? We said here happens the Calvin cycle or the light independent reactions. Okay? Now, this, as we said, is the chloroplast. Okay. Now let's first speak about the light dependent, which happens in the thyroid. And then we speak about the independent, which happens in the stroma. So here we have the dependent, and here we have the independent light reaction. Let's start. So what happens in the thyroid? How does the light come? Let's say we have this cell, this atom, okay? It has inside the protons and the neutrons, and it's gonna have outside in the shells we have electrons. One electron, it will be excited to the outer layer. It will come and sit here, and then it will absorb. By absorbing a photon, this photon is gonna excite it from place to another, from a low energy level to a higher energy level. So what happens here? This electron will excite, this photon will come and then bring it back to its place again. To do this and to come and set again in the first energy level it was in. So what happens? The photon, the electron will be excited to the outer membrane until the photon will come and then bring it back to its real place again. This is called the light absorption by the photon. Okay. However, if we speak, this is for the light dependent reaction. Where does this light dependent reactions happen? They happen in the thyroid. This excitation reactions happen in the thyroid. Now, if we speak about the independent reactions which happens in the stroma, we're going to speak about the Calvin cycle. Okay? Calvin cycle starts with an enzyme. This enzyme called the Rubisco enzyme. This Rubisco enzyme will undergo fixation or will make the carbon fix, okay, to any organic compound, and then it will be produced the 3-phosphoglycerate. This is phase 1. So here 6 ATP will be used to produce 6 ADP. Okay, this from bioenergetic look. And then G3P will be produced. While producing G3P, what will happen? 4 NADH will be used to produce 6 NADP plus, and it will also produce 6 inorganic phosphates. This G3P gonna undergo another reaction or another phase, which is the phase 2. So phase 1 is fixation. Phase 2 is reduction to make another G3P, and then it will undergo phase 3, which is called the regeneration phase. This regeneration phase will use the RUBP and the cycle will continue again. Here also, since it's the Calvin cycle, and if you remember when we do the chart, the Calvin cycle will use CO2 to produce glucose. So here, of course, we're gonna use 3CO2 by the beginning of the cycle. And the 3 ADP here, 3 ATP will produce 3 ADP. Okay. So the result of the Calvin cycle are for an ADP plus, no, 6 NADP plus is here. And how many ADP? 6 ADP here, 3 ADP here. Okay. 9 ADP, 6 inorganic phosphate, 6 NADP plus, and yeah, that's all. This is the product of the Calvin cycle. Okay. Now, so as we said, we start with 3CO2. Okay. The Rubisco enzyme will interfere. This Rubisco enzyme will allow the fixation of carbon on any organic compound When this carbon will be fixed, 3-phosphoglycerate will be produced with these products, which are the 6 NADP plus, 6 inorganic phosphate, and the 9 ADP, producing also G3P, allowing the three phases to occur, which is the fixation phase, phase 1, reduction phase, phase 2, and the regeneration phase, phase 3. This is mainly the Calvin cycle. Perfect. This is mainly the Calvin cycle. And as we said, photosynthesis is an endothermic reaction, which is the complete opposite of glycolysis. Okay. Now, we finished about glycolysis and photosynthesis. Okay. Now we're going to speak about muscle contraction and so let's start with the muscle. So skeletal, muscle anatomy. How does this skeletal muscle anatomy? We all know that the skeletal muscle basic unit is the sarcomere. So we mainly should know what is the sarcomere, which is the smallest unit of the skeletal muscle. So sarcomere as a definition, it's the smallest unit of the contractile muscle. How does it look like? Like this. If you see, we have two lines, one thick line and one thin line. The thick ones are the myosin and the thin ones are the actin. So the thick ones here, this is called the M line. Okay. This is the thick one, which is called the myosin. Smallest unit here. And you have the thin one. Okay. We have the thin, the actin. This is called the I band. Between two Z lines. This is one Z line. This is second Z line. Between two Z lines, we have the I band. Here we have the H band. And here we have the A band. Okay. So let us now zoom in this, or let's zoom in which part? Let's zoom in here. Okay. This small portion, let's zoom in and see how does it look like from inside. So perfect. So it looks like this. Mhm. Okay. So here, as we said, we have the actin, which is the thin one, and here we have the M line, and here we have these ones are called the myosin. Okay. Now, what you should memorize about all of this, that when we contract this sacromere, when we contract this skeletal muscle, what happens? When we contract this sacromere, we spoke about the M line, the A band, okay, the I band, the Z line, the H band. You should memorize that when we contract this sacromere, the M line is always constant, the A band is always constant, the I band shrinks, the Z line moves closer to M line, and the H band shrinks. Okay, so you can memorize it in this way. How I memorized it? I memorized it HIZ or high. Okay, H, I, and Z, three of them will change. However, everything else will stay constant. Okay, so H will shrink, I will shrink, Z will move closer to the M. However, the M will stay constant, will never move, and the A will stay constant. This you should memorize by heart. Okay. Okay. So now let's take the anatomy of all of this muscle, not the sacromere, but all of this skeletal tissue, skeletal muscle tissue. So first it looks like this. Yeah. So here is the sarcoplasmic reticulum. Here are the T tubules. And here we have the sarcolemma. Here are the mitochondrions. These are the myofibrils. And that's all. What happens for the muscle to contract? What is the physiology of muscle contraction? We have something called the sliding filament model. Okay, so how does this muscle contract? We have 4 phases that this muscle undergoes in order to contract. So first of all, what is the reason of voluntary or of muscle contraction? Is a nervous impulse. It's the brain that tells, or it's the nerve that tells the muscle, okay, now you can move. Okay, so what happens here? Here the neuron will come, and will sit behind the sarcoplasmic reticulum, and then the acetylcholine vesicles will go out in the cleft, and then they will sit on the receptors on the ligand-gated channels, which are here on the layer on the membrane of the sarcoplasmic reticulum, and then they will allow a kind of impulse for the sarcoplasmic reticulum as if they gave it the permission to move, and then it will start moving or contracting. Okay, how this happens I will explain now. Okay. So as we said, we start first by the by the neuron. So okay. So let's say we have okay. Before I draw this, before we do this, let me speak about the physiology of muscle contraction. So, as we said, we have 4 phases. Phase 1 is the relax phase. Phase 2 is the contact phase. 3 is the termination or contraction phase. Phase 4 is ATP hydrolysis. Okay. And the relax phase, they look like this. We have here the actin. These are the actin. Here we have the tropomyosin. This tropomyosin. And down here we have something called the troponin. Okay. The tropomyosin and the troponin, they prevent myosin from binding. Okay. Now, what happens? They can prevent or they can allow. Okay. In the contact phase, the myosin, this will look like this, as we said. And here is the M line, and here is the myosin. As we drew it, looks like this. Remember, we drew it like this. This is the myosin. So this is the myosin, and this is the myosin head. The myosin head tries to be in contact with this troponin. Okay, so it tries to be in contact with the troponin. When it's in contact with the troponin, it will change its shape. When it changes its shape, here the contraction will happen. Okay, so here it will come and bind to the troponin, and then it will change its shape, and then the contraction will happen. Now, before all of this, as we said, we have a nervous impulse. How does this nervous impulse happen? Let me explain it. So this nervous impulse, as we know that any nervous impulse to allow the acetylcholine neurotransmitters inside this neuron from the presynaptic part to the postsynaptic part, going through the cleft, we need the calcium ions, K2 plus ions. So one of the most important ions that are involved in the muscle contraction are the calcium. Why? Because let's say here we have the presynaptic part of the neuron, and here we have the postsynaptic part. So here Ca2 plus will enter. When Ca2 plus enters, this acetylcholine will start to go out by exocytosis, and they will come and sit in the synaptic cleft to be able to bind to the ligand-gated channels on this sarcoplasmic reticulum. When they sit here on the receptors, they gave a permission for the sarcoplasmic reticulum to start contraction. The moment they gave it permission, this troponin will take a calcium ion. When it uses this calcium ion, the moment it uses it, it will allow the contact between the myosin head and this troponin. When the myosin head and the troponin, they are in contact together, here what happens? The termination of contraction will happen. So here a pulling of the actin to the M line and Z lines get closer to the M line. So they start the moment that this happened, the moment that it took Ca2 plus, and then it made the myosin head in contact with the troponin. This troponin will make a kind of stop, and it will try to prevent this myosin from binding again to it. And what happens here? The M line and then the M line and the Z line will get close to each others. Okay? When does the M line and Z line get close to each other? In the third phase, which is the terminal or the contraction phase. By the end of this contraction, ATP will be hydrolyzed or broken down. We don't use ATP to contract. We produce ATP after contract. We break down ATP after contraction. So the thing you should know in the muscle contraction that ATP is hydrolyzed after contraction and not before. Okay? Why do we hydrolyze ATP? ATP is hydrolyzed. Why? Because it's used to terminate the myosin head and not to activate it. Let me write. So why does ATP hydrolysis happens? Because this ATP is used to terminate myosin head and not to activate, used to bring down this myosin head instead of bringing it up, because now we finished contraction, we have to go back to the relaxation phase. Okay, and then we have a repetition of the cycle, and that's all. This is how it happens. Okay, so as we said that we have the troponin, Ca2 plus will go out, and of course the Ca2 plus at the end, where does it go? Since the troponin used the Ca2 plus to bind to this myosin, this Ca2 plus at the end will be stored in the T tubules. So this Ca2 plus will go. Here we have the T tubules. They have sacs inside next to the cytoplasmic reticulum. They will come and be stored here in the sacs, in the caves. Okay. So this Ca2 plus is not degenerated, however, it's stored. So what happens? If we have the relax phase, the contact phase, the terminal or contraction phase, and the ATP hydrolysis. This cycle is always repeated. We start with the contraction, we end up with relaxation. First, the Ca2 plus will go and bind to the troponin. Troponin will try to bind to the myosin head. After it binded here, the muscle contraction happens. The Z line, the M line gets closer. When these, of course, the M line is constant. The Z line tries to get closer. If you see, I drew here an arrow. Because the Z line is trying to come closer to the M line, but the M line is constant, does not move all over the cycle. And then we have ATP hydrolysis. This ATP hydrolysis is used to terminate myosin head and bring it far from the troponin, not to activate it. And then the Ca2 plus will be stored in the vesicles inside the T tube. Now, from where does this muscle gets ATP? What is the number one source of ATP does this muscle get? From where does it get? What is the number one source of ATP? Let's see. Diagram shows a single sarcomere in the relaxed state, which answer describes the changes when the sarcomere contracts. One gets smaller, 2 gets smaller, 1 gets smaller. Okay, so 1 is between two Z lines, right? So since we said that what is between the two Z lines, we have here one thin one and one thin one is the Z line. Okay, one moment. Perfect. So the Z line, it shrinks, it moves closer to M line. However, 2 is the A band. And we said that A is constant, so A is constant, which is 2 is constant, and 1, it gets closer to the M line. So 1 gets smaller. When it gets closer to the M line means this contraction. It's contracting, it's getting closer to each other, these two lines. So 1 gets smaller and 2 does not change. Is it B? Perfect. Yeah, 2 is A band. A band is the first band in the spectrum. 1 is two Z lines. 1 is the first Z line and the second Z line. These two Z lines, during contraction, the first Z line, okay, and the second Z line, they try to be closer together. You know, when you contract something, when you press on something, it will come closer to each other's. How do you recognize it? A band is really easily recognized. Usually, in the beginning of the sarcomere, from the Z line to the beginning of the thick line is usually the A band. Just memorize the shape. The following events occur during a reflex response to a person placing a hand on a hot object. Myosin sites and actin filaments uncovered. ADP and phosphate ion is released from myosin head. Sodium voltage gated channels open. Myosin head detaches from binding site and actin. Calcium ions released by sarcoplasmic reticulum. So the person is placing his hand on a hot object. This means that the muscle will contract. Okay. Myosin binding sites on actin filaments uncovered. Okay. ADP and phosphate ion released from myosin head. Perfect. Why are ADP and phosphate ion released from myosin head? Because we need this ADP to produce the ATP at the end. It's hydrolysis. Sodium voltage gated channels open, yeah, because the neuron will give the impulse. When the neuron gives the impulse, this acetylcholine will go and bind to the receptors. Receptors are the sodium voltage gated channels. So the sodium voltage gated channels will open. Myosin head detaches from binding site on actin. No, myosin head will bind to the actin, does not detach. Calcium ions. So I guess 1, 2, 3. What's the answer? B. Okay. 5, 1, 2, 4. So 1 myosin, okay. 2, 3 is wrong. 5 is correct. From binding site on actin. Okay. So myosin binding sites on actin filaments uncovered. This is clear for everyone, right? ADP and phosphate ion is released for ATP hydrolysis at the end. This is correct. Sodium voltage gated channels open. I'm not sure why no. And the myosin head detaches from binding site on actin. and the last one is calcium ions, and the last one is no. So we have 4 and 3. Okay. Um, okay, I guess the sodium voltage gated channels, they will stay where they will be closed. I guess because let me make sure. Is it an IMAT one before? Let me just make sure. Yeah, it is IMAT past paper. Perfect. So why sodium? Okay, listen. Okay. After the initial depolarization caused by the opening of sodium channels, these channels undergo a process called inactivation. Inactivation involves a conformational change in the channel structure that blocks the passage of sodium ions, preventing further depolarization. This inactivation is a necessary step to ensure that the muscle cell membrane is ready for repolarization and relaxation after the contraction. So they close. Okay, they said sodium voltage gated channel. Okay, they are speaking here about the Na plus, the sodium ones. They are closed. Why? Because we have a depolarization. Okay, so when we have a depolarization, since the Na plus voltage channels must be closed. Why? Because another depolarization will happen. And if another depolarization will happen, the body will still give another impulse and another impulse to stay moving. However, if they are closed the sodium voltage channels, what happens here? They will, it's a kind of sign for the body that you're gonna repolarize again. Okay. And another one is detaches from site. Yeah, myosin head detaches from binding site on actin. We said that the myosin head binds not to the actin, to the troponin. That's why they are trying to trick you. So they said actin, but it must be the troponin, not the actin. Okay. No, wait. Myosin head detaches from binding site on actin. Yeah. It's they are speaking here. Okay, I understood what they meant. Okay, so we spoke about the 4 phases of muscle contraction. The first one is the relax phase, contact phase, termination phase, and the ATP hydrolysis. Okay, when you study this, all of them help in the contraction of a muscle. These 4 of them will help in the contraction. Means the relax phase will help. and the attachment will have, the contact will have, and this contact will have, you know? So when he speaks about the contact between the myosin and the troponin, he means the contact phase. And also when he speaks about the detach or the discontact between them, which is the termination phase, also it includes. So if he said both of them, if he said during, like if he said the myosin binds to the actin or to the troponin, and the myosin detaches from the troponin, both of them are from the steps of the muscle contraction. That's why you should take it into consideration, also the detachment, because he's not going in sequence. He said the ATP hydrolysis, which is the last step at the beginning. He said it from the first step. So from the first step, you see that he's not going into in sequence. That's why you should take into consideration the detachment and the attachment. Okay. I wanted to ask another question. Can we please have early morning questions from the document of IMAT? Sayed, I want to say something that I'm having so much, why also plus there's something that there's so much problems with the website, with the like opening the meeting, putting the meeting on the website, and with my timing. So I'm having a real problem because, you know, like now it's August, so now application documents for Italy and so much things, and especially when you come to Italy, you will see how hard it is to get your documents from Italy and to go to a place and then get them back, and you know the thing. So this is a real problem. I'm trying to balance me getting the documents, teaching. I have other work. I have studying, so much things together. So that's why I'm trying my best to give the sessions that I have to give. However, if I found myself that I have other free time, I might give. Let's say I cannot promise you, but if I have, let's say on Monday, I have a rest day. Tomorrow I have the whole day, so tomorrow I might give it morning. If you want, we can solve document. If you want, I can like we can solve it. I explain it in English, even if the question is in Italian. And if you want, you can still see the level of the document, you know. So inshallah, we will see how to manage doing these questions if you want. But don't worry, because next week we'll be done with all of the topics and we will start solving. So about solving, don't worry, we will solve all together a whole week before we end this course. So don't worry. However, if you still want from the document and these things, of course, I can manage to make an hour at least 2 times a week. There's no problem. Or from now until we start really solving, at least 2 hours. Okay, now, before we finish, I want to speak about something. So, I wanted to ask, from where does this muscle tissue, from where does this muscle is bringing the ATP? What is the source of ATP of this muscle? Does anyone has any answer? Bravo. But it's not the main source. It's the secondary source of energy. I want the main one. If it does not have that source, it will take from the mitochondria. However, if it has that source, it won't take from the mitochondria. Bravo, bravo, bravo. So, one ATP source of the muscle is creatine phosphate. If there is no creatine phosphate, it will go to the aerobic respiration, which is the citric acid cycle and this thing. Okay. If there is no aerobic respiration, it will go to fermentation or anaerobic respiration. Okay, so number one source is creatine phosphate, number two is aerobic, number three is anaerobic. From here, these ATP will be used by the muscle to contract. Okay, if you remember, we were drawing, we drew like relax phase, and we spoke about the steps. The steps are called the sliding filament model. Okay, the four steps that we spoke about. Okay. Another thing, before we continue, I want to speak about so before we end the session, I want to speak about something. Yeah, about the types of muscle tissues. So we have three types. We have the skeletal muscle, we have the smooth muscle, and we have the cardiac muscle. The skeletal muscle has locomotion, means movement, and has multinucleus. It is striated muscle. What does striated mean? Striated means looks like this. Okay. It's attached to skeleton or skeletal bones, and it is voluntary. You choose if you want to move your hand or your feet, not your mind. Its location is arterial. Location is on the skeleton, the respective location. Okay. Now the smooth muscle, it allows the peristalsis, the mechanical movement of the stomach. It is non-striated, looks like this. Smooth. It's involuntary, cannot control it. Uninucleated, and it's inside location, it's inside the intestine, arterioles, lungs. Okay. However, the cardiac muscle is striated like the skeletal muscle. It's uninucleated. Okay. So the skeletal is multinucleated, and the smooth is uni, and the cardiac is unicreated. It's involuntary. Location is the heart. And it looks like this. One nucleus, one nucleus, one nucleus. So if you want to see the histology of these three muscles or muscle tissues, you see here the skeletal, smooth, cardiac. Okay. Perfect. Tomorrow we will speak about gluconeogenesis, about the glucose balance in our body, about the urinary system. Before we start with the artificial processes like the PCR, gene cloning, and vectors, okay, and the skeletal system, we still have these. And after this, we will start genetics. After genetics, we'll be done with biology. Of course, I guess we still have some things. I will just make sure from the syllabus. I'll try to see them in the upcoming sessions. And for the chemistry, of course, we still have only some things. Also, we're gonna do them in like 2 hour session. And then immediately we will start from next week solving. Okay, so have a good night and bye bye.